Most eukaryotic cells generate a contractile actomyosin ring to separate their daughter cells at the end of mitosis. How actin filaments assemble into the ring is incompletely understood, however, mainly because it's difficult to label all the actin-based structures in a living cell without compromising actin function. Mohan Balasubramanian, from the Temasek Life Sciences Laboratory in Singapore, studies cytokinesis in fission yeast, whose actomyosin rings are thought to assemble from actin filaments nucleated by the formin CDC12 at myosin-containing nodes around the cell equator.
We were trying to make a better tool for actin, and our collaborator, Roland Wiedlich-Soldner, he had developed this LifeAct tool, so we thought, okay, let's just give it a try. And we were just delighted to see that this was far better in labeling all the actin structures that we can see with phalloidins.
Compared to other actin-binding probes, LifeAct had few phenotypic effects on cells, allowing Balasubramanian and colleagues, led by Junqi Huang and Yinyi Huang, to investigate actin dynamics in mitotic fission yeast, where, in addition to the actin filaments formed at equatorial nodes, actin cables were also assembled at non-medial locations all over the cell cortex.
And then when we started doing time-lapse imaging, we could see that these cables were growing in random directions, and when they do go to the middle, they get captured and incorporated into the ring.
The incorporation of non-medial actin cables into the cytokinetic ring was especially clear in CDC25 mutant yeast, which are larger than wild-type cells, and in yeast lacking the actin-severing protein cofilin. Cable incorporation could also be seen with other actin-binding probes, including the probe previously used to monitor actin assembly at medial nodes.
We are not saying that actin is not nucleated in the middle. We are saying that it's happening everywhere in the cell. Everything contributes towards the ring assembly, and it sort of makes sense because actin filaments are actin filaments wherever they are made. And if the -N gets captured by myosin, regardless of where it's coming from, it gets incorporated into the ring.
The researchers saw non-medial filaments join the cytokinetic ring in different ways. Many cables extended toward the cell equator, while others appeared to move wholesale toward the medial region. But what nucleates the assembly of non-medial actin filaments? Cells lacking the formin 4,3, which assembles actin cables in interphase, still form cables during mitosis, so Huang et al. Then deleted the formin CDC12, which nucleates actin filaments at the equatorial nodes.
If you take away CDC12 function, neither was actin cables assembled in the middle, nor was it assembled elsewhere in the cell. So therefore, my guess is that when the cell enters mitosis, CDC12 is activated wherever it is in the cell, and when it's active, it starts making actin filaments. And if these filaments, by chance, grow towards the middle, they get pulled inside the ring. If they grow in other directions, there is actin turnover, then they just get lost.
Fission yeast ring assembly proceeds via two different pathways, relying on either the anilin protein MID1 or the membrane-binding F-bar protein CDC15. CDC12 still formed actin filaments in the absence of these two proteins, indicating that these assembly pathways act downstream of actin nucleation. Huang et al. Then identified several proteins that move the assembled actin filaments to the cell equator for ring construction. Cells lacking the myosin motors myo2 or myo51 failed to properly compact actin cables into the medial ring, and actin filaments were scattered throughout cells lacking both of these myosins or the myosin activator ring 3, as the non-medial cables became stable and less mobile.
In animal cells also, it's known that actin can be nucleated elsewhere and transported to the middle, as well as being generated de novo. So there is more common between fission yeast and animal cells. Within our own system, we think that actin filaments are nucleated and assembled throughout the cell through the action of the CDC12 formin. And those that are in the middle of the cell, as well as those that arise elsewhere, are all bundled and compacted into a ring structure. But I'm not satisfied with the answer, and there are two things we are doing. One is to take LifeAct and do more careful microscopy and biochemistry to look at the roles of myosins, cross-linkers, and so on. The second is actually to see if we can get fluorescent amino acids into actin so that we can actually study actin itself and really, nail down actin dynamics. It's kind of interesting, right? This protein cannot be tagged in a functional form, and we still don't have satisfactory means to understand it, even though we understand a lot about other proteins that interact with it. It's still an enigma.
In the meantime, you can learn more about the non-medial assembly of actin cables and their incorporation into the fission yeast cytokinetic ring in the paper by Huang et al., published in the November 26, 2012 issue of the Journal of Cell Biology.